So we will be discussing with Dr. Danae Schulz, who received a BS in biology from Tufts University and a BM in violin performance from New England Conservatory. Her PhD work at the University of California in Berkeley focused on gene rearrangement of antibody segments in B cells. She went on to conduct postdoctoral research in molecular parasitology at the Rockefeller University in the US as well. She's currently an associate professor at Harvey Mudd, a STEM-focused undergraduate college in the Los Angeles area. Dr. Schulz was the recipient of the NIH Ruth Kirchstein Postdoctoral Fellowship, a Bill and Melinda Gates Grant Challenges Award, and 2 NIH R21 exploratory grants. She is currently a Research Corporation SciLog Scholar in the Mitigating Zoonotic Diseases Program and a recipient of a career grant, which is the NSF's most prestigious award in support of early career faculty. Danae, thank you so much for being with us.
Thanks.
We will be talking about her recent lab protocol in PLOS ONE entitled Adaptation of CUT&RUN for use in African trypanosomes at the associated protocol in protocols.io. Just to kick off, can you briefly explain what the protocol is about, how to use it, and why it is relevant?
Yeah, so CUT&RUN is a protocol that's analogous to ChIP-seq. So the idea is that you're interested in the genomic occupancy of either a histone modification, or a chromatin interacting protein. So our lab is really interested in chromatin interacting bromodomain proteins. And these are proteins that have been shown in lots of systems to be important for gene regulation. And in our system, we found that they seem to be really important in the transitions between life cycles for African trypanosomes, which is the parasite that's our model system. So we're really interested to know where on the DNA bromodomain proteins localize as parasites transition from living in the blood, where they cause a fatal disease, to living in their insect vector in the fly.
And so to help us understand the significance of this of the protocol itself, can you elaborate a little bit more on the state of the art? So what were the techniques to assess DNA occupancy when you decided to optimize the protocol, and why you had the motivation to do so?
Yeah, absolutely. So I think ChIP-seq was kind of the most ubiquitous protocol for looking at this. And there were a few things about ChIP-seq that we were not totally happy with. So the first is that it uses formaldehyde cross-linking, which some folks have shown can create artifacts in certain situations. So we were always nervous about the formaldehyde cross-linking step of that. It also requires a sonicator, which is kind of a specialized piece of equipment, and not everybody has it. And also sonicators, sometimes the DNA fragments that you get out of that can be a bit variable, so you have to keep checking it to make sure that they're the right size. And then finally, ChIP-seq protocols take a few days. And so particularly for us, our lab is staffed with undergraduates, and we wanted these students to be able to do this experiment in a short time frame. So CUT&RUN kind of gets you the same data, but in our lab, it only takes about 4 hours to run. So undergraduates can do this in an afternoon, accommodating all their complicated class schedules, and it just really streamlines the science for us. So for those reasons, we were really interested in getting it working.
I think that you have very clearly explained why previously published protocols didn't work for your interest and for your setting as well. For the user who wants to implement your protocol, what is your advice in terms of the most critical steps?
Yeah, absolutely. So, so shout out to the Hennekoff lab, who they were the original folks who published CUT&RUN, and they did an amazing job laying out a protocol in their first publication, which we definitely used as a guide. It was all excellent. For us, though, the sticking point was that the permeabilization step, as outlined by them using digitonin, just really didn't work in our hands. And so for us, the critical part of getting this working was to make sure the permeabilization was working in our system. And so I think for people thinking about adapting CUT&RUN to their particular system, the permeabilization step is really important to get right. And so what we ended up doing was coming up with a flow cytometry assay to specifically look at efficiency of permeabilization. And then we tested a whole bunch of detergents and then optimized the conditions that way. So I think if folks who are getting this up and running can make sure the permeabilization is working, then for us at least, the rest of the protocol went really smoothly as initially described by the Hennekoff lab.
And so my next question are probably two questions in one. And so if other folks would like to translate your protocol to other non-model organisms, and do you think that's where they should be playing around? So that's one question. And the second question is like, have you tried? Have you tried translating this protocol for this specific trypanosoma to a different one, for example, to Cruce, for example?
Yeah, so I think the protocol definitely could be applied to other parasites, but because the composition of our T. brucei parasites is so special, so it's covered with this variant surface glycoprotein, which is really unique to that parasite system, I would say that, yeah, probably people's efforts are going to be in making sure that for their particular cell surface, their permeabilization is working. And so what we ended up doing was we started with an anti-histone H3 antibody. And so we would permeabilize our cells with whatever condition, send in that antibody, and then send in a secondary fluorescently labeled antibody, and then just check by flow cytometry that we could see those cells lighting up. And so that would be my advice for someone trying to adapt the protocol, is just to start with that, start with a nice bright histone antibody, and check by flow cytometry that you can light the cells up after your particular permeabilization protocol, because that's actually a very fast experiment. You don't have to go through the whole CUT&RUN experiment to tell if it's working. You just go through that first permeabilization step. And then once people have that working, I would say the rest of our protocol is pretty standard. I would say that the other thing that I'd recommend folks try that we haven't tried, but is on our list to try, is cut and tag. So instead of making a DNA library at the end of the whole protocol, you can sort of make a DNA library as you're going by using this tagmentation technique. So that's next on our list to try.
So, and I remember that you mentioned that at the beginning of the protocol, you had to play with a battery of detergents to get it right, and you committed to saponin. So can you just briefly mention what else you tried and clearly didn't work?
Yes, absolutely. So we worked a lot with digitonin. That was the detergent that was originally published, and we tried really hard to get that to work. And we just couldn't get it working in our hands. And so then we turned to Triton X and P40. So we tried lots of different concentrations of those detergents as well. And then I was actually presenting at UCLA, and this like really excellent biochemist, Peter Bradley, said, why don't you try saponin? And I said, oh, I've never even heard of saponin. We've never used it. I've never seen it anywhere in these protocols, but we'll give it a shot. And it just worked like magic. So we did play with the concentration a little bit, but the signal was so much better than anything else we tried. We were really happy with it. It's very stable. In the lab, digitonin was always crashing out on us. With saponin, we make it, it's stable, it works for a long time. It's been great. The other thing that we tried was really trying to separate out the nuclei before permeabilization. And I think we also tried concanavalin beads. And through all of that, we just were never 100% happy with the separation. Like we always had some cytoplasmic contamination in our nuclei preps. And in the end, we thought, maybe this is just more complicated than what we need. Like all we really need is just to get the antibody in. And so if we can just permeabilize the parasites without a separation step, but without beads, without this and that, and the antibody goes in, that's really all we care about. And so for us, it was like faster and simpler to just permeabilize the parasites. And then at the end of the protocol, just harvest what was in the supername for those parasites.
And changing now gears a little bit, I think that we have discussed with detail the intricacies of the protocol, but I would like to know your motivations to not only optimize it for your needs, but also first publicly sharing in the open access platform of protocols.io, and then going one step further and have it peer-reviewed as a standalone methodological paper. What was the motivation behind that?
Yeah, absolutely. So we initially published this protocol in a paper that was just a straight science paper. So it was just the results of looking at these bromodomain proteins in these different life cycle stages. And then once we published that, we started to get requests asking for the protocol. Like people would email us and ask for the protocol, and we were very happy to share it. But I just thought, well, this is kind of a barrier, right? So someone has to actually get up the courage to go and write to me to get the protocol. And if it were just public, then that barrier would be lowered. So maybe someone who's not quite as confident, like a young researcher, doesn't feel good about like emailing PIs out of nowhere in the cold, could just have access to this. And then if they ran into issues with it, then they could email me with problems if they ran into them. So I liked the idea of it just being open and trying to get it out there so that as many people as possible could use it, because again, it's like it's a bit faster than ChIP-seq and more streamlined. So that was one motivation. And then the other motivation was that my undergraduates who worked on this, they worked on it for years. It was really hard for us to get it working. And so I just thought it was nice for them, like to have another like peer-reviewed publication out of all those efforts, and B, to kind of model for them that we should try to be as open and explicit as possible with new techniques that we come up with in the lab so that other people can use them. So for those reasons, we really liked that the PLOS publication supported methods that had already been published, right? So it didn't need to be a brand new thing that had never been published on. They just want it to be robust. And so we like the idea of having like a peer-reviewed publication with a really detailed protocol, because sometimes in, a straight ahead science paper is not that detailed, right? So yeah. Nice to have like all the steps laid out.
And can you share with us how the feedback that you received from the peer reviewers shaped the final version of the protocol, if any?
Yeah, so our peer reviewers actually didn't have that much to say about the protocol itself. They were kind of more focused on validation. And one nice thing that came out of peer review was somebody asked us to perform a FRIP analysis, which is basically the fraction of reads in peaks. So it's a sort of a metric for how much noise there is in your results. And I was honestly, as I was gearing up to calculate this, I was like a bit anxious. And I thought, oh, like, what if this looks terrible? I don't know what this is going to look like. But then once we calculated it, it actually compared really favorably with other folks' FRIP scores. And in fact, in some cases, it was like a bit better. And so that was a really nice validation that it was working well and there wasn't a lot of noise in our system. So that was a nice thing to come out of peer review. And then for some of the other validation things, we had actually kind of validated it in the initial paper. So we just sort of pointed reviewers there. Yeah, but they also had some like nice ideas for controls, and so we put those in, and I thought it made for a stronger paper.
And I know that this is a relatively recent paper, so I don't know how much of a guess we will have to do with this next question, but what do you think has been or will be the significance of having this lab protocol paper published for your career as an early career scientist?
Yeah. So one really nice thing about it is that I wrote this proposal for the NSF to use this cut-and-run protocol, and we had done some initial experiments that made it seem like it was working, but we hadn't done the big experiment. And so I got really lucky, and it was funded, this proposal. And so I'm just delighted to be able to actually deliver on this proposal and say, like, yes, we did the experiment. Yes, it works. And yes, we've made it like widely available. So I think, you know, having that pan out for that grant proposal will help with the next grant proposal, et cetera. So that's great. And then I think I'm always in my career just trying to support the careers of my undergraduates in the lab. And so them having this publication is also really nice for their careers. And I'm just hoping that it will help kind of smaller, less resourced labs do these types of experiments because, again, it doesn't require a sonicator or any specialized equipment. And so I'm hoping that it will help some other people's careers who might not have had access to this type of data.
And I think it has been like a very insightful conversation. Before we wrap up, is there anything that you would like to mention that we haven't touched so far?
Let's see. I can't think of anything. I would just say for folks who are getting started with it, we found that just trying to be kind of fast is important. Like, don't dawdle during the experiment. We found that if we're like sort of efficient, we move through it quickly, it goes well. If you work slowly, it goes a little less well. So that would be my recommendation. And also keeping things cold just seems to help. So making sure that centrifuges are chilled and everything's on ice helped us get the best results. And the other thing I would say is just if you run into problems, like feel free to contact me and I'd love to try and help or give you ideas.
Oh, that's very generous. Well, thank you so much for your time again.
Thank you.